Hello, my name's Mike Ward. I'm the head of content at Informa Pharma Intelligence. We're here in Vienna at the BioEurope Spring Meeting where pharmaceutical companies, biotech companies and investors all meet to discuss the trends that are impacting the industry and also looking for opportunities, whether that be for partnering or whether that's in fact for investment purposes. One of the areas that has sort of gathered a lot of sort of interest from investors in recent years are companies who are focusing on RNA, but sort of different types of RNA. I'm joined by Roel Schaapveld, who's the CEO of InteRNA, which is a Dutch-based biotech, and you're focusing on microRNAs. So, Roel, what's so important about why microRNAs in that sort of that family of RNAs that one could pick on?
Yeah, so microRNAs represent a subclass in this RNA therapeutics field, and they are actually the master regulators in your body, regulating the translation from your genetic code into functional proteins by regulating the messenger RNAs, so they can bind to the messenger RNAs and thereby inhibiting the translation to protein. You have 20,000 genes in your body, but only 2,000 microRNAs. So each microRNA can regulate more than one messenger RNA at the same time.
Okay.
And we feel that especially in multi-gene diseases like cancer, we focus on cancer, that if you hit only one target, you don't have the long-term efficacy. And that's why you see the developments in the oncology field with all kinds of combination therapies, with kinase inhibitors, but also with the checkpoint inhibitors.
Yeah.
And our lead candidate actually has a dual mode of action by interfering in pathways in tumor cells, but also modulating the tumor microenvironment and basically giving a 1-2 punch to the tumor by eliciting also a long-term T-cell mediated immune response.
Right, and sort of the approach, is it the microRNA that you're actually using as the therapeutic molecule?
Yeah, so we started off, and that differentiates us from other microRNA companies in the field. We started off with functional genomic screening. So we had every microRNA in a lenti-vector system to allow us for high throughput functional screens and cell-based assays. Gave us immediately insight in potential function of all these individual microRNAs, and then we took it from there to further validate them. And then in cell lines, but later on also in vivo models, of course. Now we are at the stage that we have done dose range findings in rat and non-human primates, and now we are ready basically for the formal GLP tox we need to do for the authorities to file our first CTA for our first clinical trial later this year.
And that clinical trial would be, have you already picked out the indication you're going to go for?
Yeah, so in our target product profile, initial aim in the 1B/2A trial is a more basket-like approach. One basket with hepatocellular carcinoma patients that are resistant to sorafenib. Sorafenib is the only approved first-line treatment for liver cancer that cannot be removed by surgery. These patients are really sick, and sorafenib is a dirty drug in the basis. And then the other basket will be chemo-resistant triple-negative breast cancer. Wow. And in both fields, you see also that the checkpoint inhibitors have not yet arrived. In HCC in second line, they are there, but if you look at the efficacy, you can put some question marks behind it. In TNBC, they are not approved. Some countries do provide actually off-label to chemo-resistant triple-negative breast cancer patients.
So you mentioned sort of the checkpoint inhibitors, and these days there's a lot of focus on developing combinations. So you get your PD-1 or your PD-L1, and then you sort of bring in other molecules that are involved elsewhere in that sort of oncology cascade. Yeah. Do you sort of envisage the microRNAs would be part of that arsenal, or could they be developed as products themselves?
They can certainly be positioned also in combination, but with our lead candidate, with this dual mode of action, what we see in the biology is that it can make tumors from cold to hot by recruiting CD8 positive T cells, but the nice observation, what we see is also in parallel of downregulation of the immunosuppressive T regulatory cells. There are other RNA modalities out there like RIG-I, toll-like receptor 9 agonists, CpG oligos, that can also make tumors from cold to hot by recruiting CD8 positive T cells, but they cannot downregulate the Tregs at the same time. So these modalities need to go in combination with checkpoint inhibitors, whereas ours potentially can go for monotherapy potential. That's also what we would like to test in our first trial.
Right. And scientifically, what are the challenges you still need to get over to reassure any skeptics that this approach is scientifically curious, but actually it's never going to work?
Yeah. So the first challenge that we needed to address was drug delivery. Drug delivery for RNA therapeutics, that's everybody's talking about in the field. So it took us a couple of years to get there, but we added to our screening platform another platform to screen for formulations, and we were able to identify a formulation that can bring our cargo beyond the liver after IV administration to tumors elsewhere in the body. So we have addressed that issue. Then the second thing is, of course, what's the MOA? It's not a unigene single target biology. Yeah. It's a pleiotropic effect across pathways, and we're getting more and more insight in that as well on our lead candidate mainly, how it exactly works, what pathways are affected in the tumor cells, how it is eliciting the T cell immune response. Our lead candidate can also affect the adenosine pathway, basically also there playing a role to inhibit the evasion from the immune system in that way. So it's all combined, but still specific on specific messenger RNAs in these pathways.
Yeah. And any discussions you've had with regulators? I mean, because I could have thought that less precise approach might sort of ring some alarm bells for regulators?
Yeah, basically, MiRNA, for example, is already in phase 2 with an anti-MIR and also with a mimic. So basically, they set the field also for the regulators, and we are not expecting their major hurdles actually.
Okay, and so the financing to date of the company, what does that look like, and have you got enough runway to be able to sort of meet some of these early stage goals that you're looking to?
Yeah, we are VC backed by Aglaya Oncology Fund and InCAF Capital with seed and Series A. We actually recently initiated efforts for Series B funding that should bring us to proof of concept in this 1B phase 2A, and also bring a second program up to IND filing by the end of 2021. Okay. It should be a double-digit round.
And the actual ambitions for you to move these further forward. I mean, how far can the company pursue these independently?
Certainly up to the proof of concept in 1B/2A, and then we will judge the data coming out of the trial. You have options then. You can go for Series C as a pre-IPO round to fund your phase 2B trial, or we also think that you may have an M&A opportunity on the back of those 1B/2A data for the lead candidate at least. So.
Okay, well, Roel, thanks very much for stopping by.
You're welcome.
Great.
Thanks.
Cheers. Thanks.